Welcome to Sekisui Xenotech's webinar, To See or Not to See, CYP2C Induction Studies for Successful Preclinical Risk Assessment. I'm pleased to introduce our speakers for today's webinar, subject matter expert Becky Campbell and our technical support manager for services, Dr. Andrew Taylor. Becky Campbell is a principal scientist in the program Oversight department and has been with Sekisui Xenotech since 2001. She obtained her Bachelor of Science in Biology from the University of Central Missouri and studied toxicology and pharmaceutical sciences at the University of Missouri, Kansas City School of Pharmacy. Becky has extensive experience in vitro CYP induction studies, regulatory compliance, and project management. As a principal scientist in scientific operations, she is responsible for the conduct and oversight of non-clinical drug interaction-related contract studies in our program Oversight department. Dr. Andrew Taylor is our technical support manager for contract services. Andrew received his Ph.D. from the University of California, San Diego, Scripps Institution of Oceanography. He joined Sekisui Xenotech as a research scientist in 2017, serving as study director in non-clinical drug interaction contract studies and specializing in drug transport and drug metabolism studies. As technical support manager, Andrew provides a bridge between clients and our expert scientists, as well as oversight and guidance in client relationships to ensure research needs are being met. With that, I'll hand it over to Andrew and Becky for today's presentation.
Thank you very much, Madison. Let's go ahead and get started. And thanks again for joining us for our presentation on the CYP2Cs and CYP2C induction. Over the years, we get a lot of questions about the CYP2C induction studies, and there does seem to be some mystery or a lot of questions behind it. So hopefully we can unveil those mysteries, and by the time this is over, everyone should have a really good understanding of when, why, and how to conduct CYP2C induction studies. So just a brief outline of what we're going to go over today. At the beginning of the presentation, I'd like to just briefly give a little bit of background about CYP induction studies in general. So why is measuring CYP induction important? What are the mechanisms of enzyme inductions that lead to drug-drug interactions? We'll go over some real-world induction drug-drug interaction examples, and also just go over some basic terminology that's used in enzyme induction studies. Then we'll follow up looking at what the regulatory guidance recommends when it comes to conducting in vitro enzyme induction studies. And then Becky will join me and we'll discuss some of the common questions that we received over the years on CYP2C induction. So moving on first, why is measuring CYP induction important? As many of you I'm sure are aware, it's the cytochrome P450 enzymes are responsible for a majority of the metabolism that occurs. This pie chart here comes from a paper by Cerny and others that was conducted in 2016, where they looked at all of the drugs that had gained approval between 2006 and 2015. And group them according to the enzymes that metabolized them. And as you can see, more than half of the drugs that were approved in this time period are metabolized by cytochrome P450 enzymes. So looking at drugs interactions with this family of enzymes is really important in determining the drug safety and assessing drug-drug interaction possibilities. So the general mechanism of how enzyme induction, specifically CYP induction, can lead to drug-drug interactions. In this little cartoon shows, let's just say this person is taking drug 1, and drug 1 happens to be metabolized by CYP3A4. Now, when they're just taking this drug alone, the drug is doing its thing and being metabolized, and this person is having normal metabolism of drug 1. They're getting their normal intended exposure to drug 1, and drug 1 is having its intended efficacy, and it's being cleared just as intended. So everything's going as normal. However, if this person were to start taking another medication, in this case drug 2, which happens to be a potent CYP3A4 inducer in this example, the second drug will lead to an increase in the amount of CYP3A4 present. So it's inducing that enzyme, and there's more there. The increase in the CYP3A4 enzyme and most likely increase in CYP3A4 activity will also lead to an increase in the metabolism of drug 1. In this case, that will cause a reduced exposure for drug 1 and reduced efficacy of that drug. Now, keeping that mechanism in mind for that drug-drug interaction, we can give a couple real-world examples of this. Now, one of them, the first one, I'm sure that we're all familiar with as we heard about it back in our high school health education days, and that's the interaction between oral contraceptives and rifampin, or birth control and antibiotics. It's just like the mechanism that I just showed. So oral contraceptives work by increasing plasma hormone levels, estrogens and progestins. These estrogens are metabolized by CYP3A4. However, rifampin is a very strong CYP3A4 inducer. Therefore, if someone is taking oral contraceptives and then starts taking the antibiotic rifampin, it would lead to an increase in CYP3A4, thus leading to decreased levels of plasma estrogens and decreased efficacy of the contraceptive. Now, while this drug-drug interaction is not necessarily considered life-threatening, I think we could all to some drastic life-changing consequences. Another example is the supplement St. John's wort. Just like rifampin, St. John's wort is a CYP3A4 inducer. So taking St. John's wort can reduce plasma concentrations of other drugs that happen to be substrates of CYP3A4. So just to go over some really basic terminology that's used in enzyme induction studies. The first term is EC50, and this is the concentration of the drug that gives the half maximal response. And conceptually, you can think of an EC50 as being similar to an IC50, but it's looking at the response of the production of more enzyme instead of inhibition. EC50s are a concentration, so they'll always have some sort of concentration units attached to them, such as micromolar or some sort of weight per volume unit. The next term is Emax, where E is the effect at drug concentration C, and Emax is the maximal effect at a high drug concentration. And this is when all the receptors are occupied by the drug. Emax is a fold change, so it's compared to the vehicle control. The last term that's used a lot in enzyme induction studies is prototypical inducer. And a prototypical inducer is a compound known to induce a particular enzyme. You can think of it like a positive control. So for example, rifampin is a prototypical inducer of CYP3A4. So enzyme induction is a receptor-mediated response to drugs. The drug enters the body and then enters the cell where it binds to a nuclear receptor. Now this triggers a signaling pathway which leads to gene transcription of a given enzyme. The gene is transcribed to messenger RNA, and the messenger RNA is converted into protein. The protein is folded and moves into location where it becomes active. Now, since all these processes have to take place, the time frame of enzyme induction is on the order of hours to days, as opposed to enzyme inhibition, direct inhibition, which can be seconds to minutes. So to see the effects of enzyme induction, it takes a much longer time period. Now down here is just illustrating that these are some of the nuclear receptors, such as AHR, it's a receptor that activates the induction of CYP1A. CAR activates the induction of CYP2B2. But notice here that there's crosstalk between certain pathways, such as the CAR and PXR pathway, where the PXR pathway can activate induction of CYP3A, but can also do it for CYP2B. And this is just to point out that induction is a lot more complex, and it's not necessarily a one-to-one relationship between the nuclear receptor and what particular enzyme is going to be induced, which is exactly what leads us to the topic of CYP2C induction and when we need to address it. So the 2C CYP family is induced by the PXR, the pregnane X receptor, which also activates the induction of CYP3A4. So both of these isozymes are activated by the PXR pathway. So when do I need to assess CYP2C induction? The FDA 2020 guidance for industry states that if no induction of CYP3A4 enzymes is observed, evaluating the induction potential of CYP2C enzymes is not necessary because both 3A4 and 2C are induced via activation of the PXR pathway. And this makes sense. If we don't see induction of 3A4, CYP2C is not being induced. However, if the investigational drug does induce CYP3A4 and the results suggest that a clinical study is warranted, the sponsor should evaluate the potential of the investigational drug to induce CYP2C. So again, when do I need to assess CYP2C induction? If we get a positive response for CYP3A4 induction, we need to evaluate CYP2C, 2C9, and 2C19 for their induction potential. Now this little plot down here is just an example of some data that's obtained from an induction study. In this case, it's for CYP3A4. And just to orient you to the plot, the y-axis are the different concentrations of the drug being tested, along with the vehicle control, the negative control, and the positive control. And the x-axis is the fold change, again, and that's compared to the vehicle control. And you can see in this example that we see some really beautiful concentration dependence with the induction of this enzyme. As the concentration of drug is increased, there's a greater fold change. And the positive control down here at the bottom, the prototypical inducer that's used, shows that the test system is functioning properly and as expected. So CYP2C induction study design, what's different? Basically, the study design of looking at CYP2C induction is pretty much exactly the same as what we would do in a normal induction study, where you're just looking at CYP1A2, 2B6, and 3A4, mostly, and I'll get to this at the end. But the study design that we carry out here at Sekisui Xenotech is robust, and it meets the requirements of all three regulatory agencies, the FDA, the EMA, and Japan's PMDA. And the guidelines should recommend that induction studies need to be conducted in plated hepatocytes using a minimum of 3 donors. We incubate our systems for 72 hours with test article that's treated every 24 hours. The study design needs to include spent media analysis, which is looking at how much of the drug is in the media at the end of the experiment over several different time points, just to assure that the cells were exposed to the level of compound that you intended to. And the experiments also need to include a cytotoxicity assessment to make sure that the drug isn't killing the cells. Now here's where it gets a little different than standard design. So in the standard CYP induction experiment, the main endpoint to look at is messenger RNA. And that's what we use for CYP1A2, 2B6, and 3A4. However, the endpoint that we recommend to look at for the 2Cs is actually enzyme activity. Now, this is due to an industry-wide known phenomenon of poor CYP2C19 induction when using messenger RNA as the endpoint. The exact mechanism for why this occurs isn't known, but it could be something like the messenger RNA for this particular CYP2C19 isozyme doesn't amplify well, but the exact mechanism isn't known. That's just a hypothesis. So because of this, here at Sekisui Xenotech, we recommend that the activity for all 2C enzymes be included. So just across the board, if you're following up with a 2C assessment, we recommend just looking at activity for all of them. Okay, so that's just my brief background about CYP2C induction studies in general. And at this point, I'd like to bring Becky Campbell into the discussion. Are you there, Becky?
Yes, thank you, Andrew.
Oh, you're welcome. As Madison mentioned, Becky is by far the subject matter expert here at Sekisui Xenotech for CYP induction studies, and she's been working with CYP induction, gosh, for close to 20 years. So Becky, I'd just like to get some of your thoughts and on some of these questions that we get asked a lot. The first question is, why do you recommend looking at activity instead of messenger RNA for CYP2C induction? And I know that I just briefly touched on this, but I'd like to get your thoughts as well, a little more in depth.
Yes. So as Andrew has mentioned, particularly for CYP2C19, we cannot get a reliable response with the prototypical inducer of rifampin. Oftentimes for 2C19, it is we get results that are under twofold with the prototypical inducer, which is not ideal. For the other 2Cs, ideally we would like to have a fold induction of 6-fold or greater for the prototypical inducers. And for the other 2Cs, 2C8 and 2C9, it's not always that way with mRNA. Activity is much more reliable and more robust in getting a response with the prototypical inducer.
Gotcha, great answer. And so another question that we get a lot, could you comment more on how 2C induction results may result though in data looking like it's downregulation instead? Again, this is something we get when using activity as the endpoint, or does this happen with messenger RNA as well?
Another great question. You can have a downregulation or what looks like a decrease in activity or mRNA, either one. And it's really, you have to look at the data on a case-by-case basis and evaluate what was going on in the culture as well. If your test article happened to cause some cytotoxicity at the higher concentrations, well, then you would likely see a decrease in activity and mRNA. If there is inhibition of the cytochrome P450s with your test article, you can see activity that looks like there's no increase or there is a decrease because of the inhibition and the binding of the protein so that it can't turn over the substrate for activity assays. That is the reason why the guidance agencies do recommend mRNA. So it's really nice to have a full picture of both activity and mRNA for the 2Cs, with the exception of 2C19. You can also have mRNA that declines with increasing concentrations, and that may be a case of suppression of the mRNA.
Interesting. So, having both, of course, gives you a more robust data set, right? Because, then we can look at both mRNA and activity. But you also say, we need to look at the big picture, between the different donors. Can you give an example, like what would happen if only one donor met the criteria for induction, say it had its mRNA greater than 2-fold change, but the other two didn't?
Both guidance, well, I shouldn't say both, but the EMA and the FDA, which is where I think most of our studies get submitted to, they do require worst case scenario discussions. So we can emphasize that it was only in one and not the other two, but we still have to discuss, each individual donor and the response.
And also looking, like you said, for signs of maybe cytotoxicity in the other ones, and just
yes,
approaching it holistically and looking at all the data available.
It's good to look at the trends between the 3 donors and keep in mind that they are, 3 individual donors.
Right. Oh, that makes sense. Okay, another question that comes up a lot. And do we typically save the cell lysate for the potential need to analyze CYP2Cs in case there is a positive hit on CYP3A4? This is so that we don't have to do another separate hepatocyte incubation. Right.
So we can absolutely, if we have the cDNA and mRNA still frozen after a induction study of 1A2, 2B6, and 3A4, and there was a positive 3A4 was positive with induction, then we can pull that mRNA out or the cDNA out and proceed with looking at the mRNA results for 2C8 and 2C9. 2C19 is, again, a separate story. And so we would recommend to proceed with activity. We don't currently recommend performing the substrate activity assays and then saving that supernatant or the cell lysates from that to look at 2C19 activity at a later date, because we just don't know about the stability of how long that those metabolites are stable in the supernatant.
So like in this scenario, I ran my standard CYP inhibition assay and I saw, 3A4 induction. So I went back to the lysate that we saved and I got the mRNA for 2C8, 2C19. But then I'm still going to have to do a separate incubation to look at activity for CYP2C19, right?
Correct. We can look at the mRNA for 2C8 and 2C9, but we would need to do another culture period to look at the activity of 2C19.
And since I'm following up with another culture period to look at 2C19 activity anyways, should I just go ahead and look at 2C8 and 2C9 activity as well at that time?
Absolutely, because you'll get a better data set and a more robust data set to go to the agencies with that tells more of a clinical, I can't say clinical, but more of a real life perspective of what's going on, because the mRNA can be induced, but that doesn't mean that the protein is overturned as quickly, and so that the magnitude of induction in the protein may be less than the mRNA.
And at that point too, just adding on the activity of the two enzymes isn't going to increase the cost much, because most of that cost is in doing the incubation and getting the hepatocytes for the 2C19 activity.
Absolutely. The inducible attaching plated hepatocytes is generally one of the biggest costs of hepatocytes or of induction studies, because they're such a precious resource.
Oh, that makes sense. Yeah, and then I guess it's kind of sad if you have to go and spend that extra money, but you are getting a much more robust data set, and also the guidance tells you to.
Yes.
Okay, and another question that we do get asked a lot, and this is both in regards to just the normal CYP induction study with 1A2, 2B6, and 3A4, but can we use correlation method for 2C enzymes to understand the induction potential?
This is a great question. The correlation method is really what we call relative induction score, and it does require treating the hepatocytes with non to mild to moderate and strong clinical and in vitro inducers. So you're looking at like 7 to 8 compounds, 5 to 7 concentrations, because you have to get an EC50 for each one of those inducers. And so it's really like running 3 to 5 induction studies all at once.
Wow.
So our hepatocytes that we use for the induction studies at Secretary Xenotech do have a relative induction score for CYP3A, but we currently don't offer that for the 2Cs. Could it be? Absolutely. You could use correlation method for the CYP2C enzymes as well, but we haven't developed that in-house because, again, it uses a lot of hepatocytes to do that with. And I think if you're looking at 3A4, if you have 3A4 induction and look at the correlation method for that, that's probably going to be similar to the 2Cs. And I think 3A4 induction in the clinical setting would kind of trump, the 2C as well.
That makes sense. Yeah, that does sound like a lot of work to do this the relative induction score. How often for the 3A at least do we get asked to do that?
We've seen an increase in the past couple of years for that request to use the relative induction score to classify 3A4 induction. So it's probably up by about 20%.
Wow. Interesting. And also another question that we get a lot, and again, this can relate to both just 2C induction or induction of any of the CYPs for that matter. But what would be the effect of a less than 2 times twofold CYP2C induction? So I think, you know, when people ask this question, they get their results and they get a 1.8 fold change or 1.9. You know, it's right there. So you ask yourself, okay, do I round this up and it's 2 and call it an inducer, or is it just strictly by the rules? Nope, this is under 2. We're good. What happens in this situation?
This is kind of a tricky question, and really exemplifies why induction at face value seems really simple. You just treat cells and look at the outcome, but can become very complex when trying to discuss the data and interpret the data that we have at the end. So it's case by case, donor by donor, and looking at the big picture again. We would look at not only, you know, we would look at each donor and see if there was a concentration dependent increase, and maybe one donor has greater than 2 while the other one doesn't, but it might be but you might still see a concentration dependent increase. It just doesn't go above 2-fold. And then you also have to keep in mind that the guidance agencies also classify induction with 20% effectiveness compared to the prototypical inducer. So if you have a less than 2-fold increase and with your test article, but it's still like above 20% compared to the prototypical inducer, it still has to be discussed as a potential inducer. So it's really case by case. And that's why one of the reasons we do still emphasize using activity for the 2Cs, because the 6-fold cutoff that we have for 1A2, 2B6, and 3A4 is where that 20% comes into play.
Gotcha. So yeah, you know, like you said, with a lot of these answers, it sounds like a lot of it is looking at the big picture, seeing what the effect is on all 3 individual donors. You know, are they showing a similar response, which I guess makes it a little easier if it happens to be a concentration dependent response, as opposed to just maybe one outlier at one of the concentrations, or taking into account cytotoxicity or like you said, activity, and also comparing it as a percentage to the control.
Yes.
It just illustrates, like you said, you put it really well when you said, you know, these can be deceptively simple, but then kind of difficult to interpret at times.
Yes. Because you can also, I mean, we have had cases where all 3 donors did something a little bit different. Like one may have a really pretty concentration dependent increase, while another one, there's nothing going on, and another one that is kind of bumping up and down right at the 2-fold line.
Well, the joy of being a scientist working with biological systems.
Yes, you have to remember these are whole cell assays that are very complex during the culture period.
Right. All right, so, you know, in conclusion, just to wrap this up and demystify the CYP2Cs, basically, if CYP3A4 induction is observed, the guidance requires the sponsor to follow up with looking at the 2Cs induction, either in vitro or in the clinic. You know, based on the conversation with Becky and having some more information, if you're going into an in vitro CYP induction study and you have any prior knowledge of the compound might potentially be a 3A4 inducer, it's probably best to include the 2C induction in the initial study design, as this is going to save both time and cost to be able to do everything up front and not have to go back and do that second treatment just to look at 2C19 activity. And while messenger RNA is the preferred endpoint for the main CYPs in the induction studies, 1A2, 2B6, and 3A4, when looking at the 2Cs, it's probably a better idea to go ahead and look at activity as the endpoint. And again, just because for whatever reason, the 2C19 mRNA just isn't a very good, reliable endpoint to determine CYP2C induction. And as Becky mentioned, if your budget allows and you want a more robust data set, it's probably best to look at both endpoints for all the CYPs. So this was just, again, focused on the CYP2Cs and 2C induction, but for more in-depth information on CYP induction studies or any other in vitro DDI studies for that matter, I really encourage you to go take a look at our beautifully designed new website at www.xenotech.com. On the website, you'll find lots of informative videos, webinars, blogs, manuscript posters, all sorts of great information relating to in vitro DDI studies. And if you have any questions at all on this webinar or anything else, please reach out to us through the Contact Us tab on the website, and we'd be happy to talk to you and answer your questions. And on that note, I'd like to thank you all for your attention, and we can open it up to questions.
Thank you so much, Becky and Andrew. While we wait for questions, I'd like to take this opportunity to mention the information about our enzyme induction and other contract services are available on our website. We also provide test systems for in-house assays and scientific resources relating to ADME and DDI. And that includes published papers, posters, other webinars, and more. And all those are available in our scientific resources section of our website. Additionally, if you'd like to be notified about future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding today's presentation or any of the Sekisui Xenotech contract research services or products, please feel free to ask them in the post-webinar survey or to contact us through our website. And I'll just go ahead and hand it back over to you, Becky and Andrew, so you can start taking some of those questions.
Yeah, thanks, Madison. I'll start with the first one. We'll just go in order. And that is, what do we use as a negative control for 3A induction?
So we use flumazenil, and we actually use it for all 3 CYPs. So it's only one extra treatment group for all 3 CYPs. Oh. And it's a clinical and an in vitro non-inducer.
Okay. If your compound is highly metabolically liable in hepatocytes, what is the best way to interpret the induction data?
So we started looking at how much test article is still in the cell culture media after that 3rd day of treatment over a time course from 0 to 24 hours post the last treatment.
The spent media analysis, right?
Yes, the spent media analysis. That's what we've referred to it internally. And we've seen it all over the board. We have seen the compound be completely gone at that 24-hour time point. So after 24 hours, well, actually it's the 3rd day, so the 72 hours of exposure, and there be no induction. And we've seen it still mostly all there present, and there is still high induction. So currently, we don't recommend interpreting your induction data based on how much test article is still around in the test in the spent media.
Interesting. Okay. Can you get only one CYP2C isozyme induced, or is it all or none?
Actually, you can. We do in some of our 2C data where all 3 enzymes are evaluated. We've seen test articles that only induced 1 or induced all 3. I think it has something to do, of course, it's the class of drug and the population that you're going into as well eventually, but there's also some like magnitude of induction involved with the 2Cs versus the 3A4. Mm-hmm. And sometimes I think that you can get a greater magnitude of induction with 3A4 than you can the 2Cs.
Okay, we mentioned a phenomenon where increased substrate concentration can lead to mRNA suppression. Is this of any consequence or concern?
If you have a test article that is causing mRNA suppression, and we do see that occasionally, you can get questions from the guidance agencies, and I don't know of a consequence in a clinical setting. and it's really where you need to take a step back and look at your big picture, your whole data set to be able to determine if it's a concern or not.
Mm-hmm.
It can't come from just one in vitro study.
Right. And I guess this is a case too, where if this happened in your in vitro study, it might be nice to actually have some activity data as well.
Yes. Yes.
To kind of tease it out.
We have seen, I mean, we've had drugs that we worked on approved that had, that wasn't necessarily suppression. It was obviously, it was an oncology drug and there was a lot of cytotoxicity, but you did see a concentration dependent decrease even prior to where the cytotoxicity picked up.
Mm-hmm.
And it was approved. Doesn't mean they didn't have questions and it wasn't a concern of the agencies, but I know the drug is on the market, which is always nice to hear.
Right.
If that helps.
Do you have to determine an EC50 and Emax value to confirm concentration dependence?
No, you do not. I mean, sometimes you have 3 concentrations that increase at the very end of your concentration curve, but the mathematics of the EC50 and Emax calculations just do not like it, and it's very unhappy and won't get you that EC50 or Emax value, which can be a little bit frustrating because those values are used further in like the mathematical modeling that is done to determine, if you're at risk for a clinical DDI, such as the R3.
Right.
You can still have a concentration dependent increase and not be able to determine an EC50 and Emax.
That makes sense. Similar to like if you're looking at an IC50, but for whatever reason can't get the concentration high enough, say to solubility, right, to reach greater than 50% of inhibition and then be able to calculate the IC50.
Yes.
If interpretation of induction is based on nominal concentration, what is the value of looking at the concentration of drug in the well? In other words, how do you use the measured drug concentration in the interpretation of induction data?
This is a fabulous question.
Yes.
It is. And the answer is we don't. We currently don't. I think there's the induction test system is a very complex model. And like I mentioned previously, we've looked at the concentration. Well, for one thing, you treat once a day for 3 separate days. So we're only measuring that concentration on that last day because the guidance agencies, that's all that they require.
Mm-hmm.
And we've seen induction with no test article remaining, and we've seen induction with a lot of test article remaining. So it's a model that, like I said, is complex. I think at the very highest hypothesis, if you had a drug that disappeared rapidly and you still had a lot of induction, or even if you didn't have much induction, you might be asked to go back and treat more than once a day, even in the in vitro system, to evaluate the potential for induction. But it's complicated. And probably not a straight answer.
And how often have you seen that occur?
We haven't been asked to treat. We've only been asked to treat more than once a day, maybe on a handful of occasions. And I think it also has to, what comes into play is how your drug is going to be dosed in vivo as well.
Mm-hmm. Again, so some prior knowledge there would really help in study design.
Yes.
Yeah.
An interesting thing about spent media, if there is cytotoxicity, then it's bad enough that it impacts the induction data. So like you see a decline in the induction at the higher concentrations. Sometimes you do see it in the spent media as well. Like you'll see a pattern of disappearance over the concentrations, and then all of a sudden it jumps back up to where you don't have much metabolism of that test article.
Oh, that's really interesting. I didn't even consider that. So again, looking at all your data and looking at the big picture to try to make an interpretation of all this.
Absolutely. One of the questions is, by guidance, 48 hours is allowed. Why do you recommend 72 as standard protocol? And that's a good question. We do have some in-house data that shows for some CYPs, it is actually better to go out that 72 hours, even if 48 is allowed. And we have in the past, we can always shorten that treatment time to 48 hours if we know upfront that maybe there's a potential for cytotoxicity, or if there's another reason. We've even done as low as 24 hours. It's not recommended, but if there's good evidence that the cytotoxicity is not going to be able to allow us to go as high as we need to for your particular test article. So it's really a case-by-case basis. If 2C8 and 2C19 mRNAs are negative, isn't it necessary to evaluate CYP2C19? Yes. If you have CYP3A4 induction, you should look at all 3 of the 2Cs. However, we would still recommend looking at CYP2C19 activity rather than mRNA, because the results from the mRNA with a prototypical inducer often being below 2-fold for CYP2C19 mRNA complicates it. And so even if you do have a negative response for 2C8 and 2C19, you still look at 2C19, but again, preferably by activity.
Okay, how specific CYP2C substrates are, and is it really CYP2C induction being measured by activity assays, or partly CYP3A induction? So
Another great question.
Yeah.
Yeah, our CYP2C substrates are specific for the 2Cs. They're substrates that we use not only for induction, but also for inhibition in our inhibition assay. So we do know that they are specific to the 2Cs.
All right, well, that was the very last question. So thank you, Becky. And thanks to everybody for attending.
Yep, that wraps up today's session. Thank you so much to all of our audience members for taking time out of your day. And thank you so much to Becky and Andrew for a great presentation. Hope everybody has a great rest of their day.
Thank you.
Thank you.